In physical science, students learned that the atom is the basic building block of all matter. And now in life science, they should discover that the cell is the basic building block of all life. That all living things are made of smaller little structures, which we call cells. To be able to see these cells is very convincing when it comes to understanding this. And just as in physical science, the most valuable piece of equipment is the balance, in life science, the most valuable piece of equipment is a microprojector, especially at the primary level, because with a microprojector, you can show cells to the whole classroom, and this one piece of equipment can be used by every teacher in your school. In fact, you may already have one of these that's been collecting dust for years. If not, I recommend you get one. The obvious advantage of a microprojector is that the students aren't looking directly in their own microscope, so you know that they're seeing what they're supposed to see. Plus, especially at the primary level, where motor skills are a problem, you can be doing all the focusing and adjusting. Now, a microprojector is really just an inverted microscope. We have the light source at the top, then we have the stage where the slide goes, and then we have the objective lens. That's the lens that's closest to the object, and this particular one has 3 objective lenses. And then where your eye would normally be in a microscope to see something, we project it. You can either project it directly down below onto a piece of paper, or you can put the mirror into position and project it off onto a wall, perhaps 10 feet away. Or actually, this Kinevision microprojector will move upward like this, and you can shoot directly onto a wall or use the mirror and shoot it onto a ceiling if you want. So it's really a very versatile piece of equipment. And incidentally, this is a Kinevision X1000. Operating this microprojector is not difficult. Once you see how to do it, and once you work with it yourself, you'll see that it's very easy. First thing we'll take a look at are these 3 little turret objective lenses. Each one has a number on it. One says 6.5, one says 10, and one says 16. The lower the number, the more magnification. So if you want a lot of magnification, you set it till it clicks on the 6.5 lens. If you're in between any of these 2 lenses, you're not going to get any light to come through here. So you want to make sure it clicks into position. So I'll choose the 6.5 lens. The next thing we have on this microprojector is we notice that this rotates down here, and we have some letters over here on this side. The first letter I see is a C. That stands for clear. And that means that if I looked underneath here, I would see that there's a circular part here and there's no lens in it. It's clear. So the light comes through the top, goes through the slide, goes through the high power objective lens and projects directly straight down onto the table. Or we could use the mirror once again and project it on a wall. If I move it one more turn, I get to A. That I like to think of as amplify. And if I look in there this time, I see a lens. This lens is a 5 power lens, so we're magnifying our image 5 more times, so we get even more magnification down here. The next turn I find here is an E. That stands for eyepiece. That's this one here, and this is a 10 power lens, and this will also project more magnification. But each time you increase the magnification, you're losing light because you're going through more lens, more glass. And as you will see in your classroom, that the higher power you go, the dimmer it gets. You could try and project it onto the back wall of your room, but it would be so dim it would be difficult to see. That's why it's important also, once again, to be able to black out your room. Pull the blackout curtains, make your room as dark as possible. The final position that we have down here is the letter P, and each one of these once again clicks into position, and you have to be clicked in or it's not going to work properly. The other thing to make sure it doesn't happen is this mirror doesn't get pushed into position, because that can happen too and you wonder where your light went. But if we keep the mirror out of the way, we run the light through here, and in P we're talking about polarizing. Now This can be used in physical science when you start looking at minerals and polarized materials. And just to give you an idea what the polarizing does, we have a polarizing filter down here. We also have to have the other polarizing filter into position. That's this one at the top. And this one at the top rotates. You can spin it with your fingers. And it's like two polarizing lenses, and you're actually changing the particular types of light waves that are coming through materials. And we're going to take a look at just some cellophane, just regular cellophane from a cigarette wrapper. We'll put the slide on the mechanical stage, and incidentally, a mechanical stage is a nice little gadget to have. It allows you to, by turning knobs, adjust the actual location of different parts of the slide without having to move the slide around. What we're going to do in this case is we're going to project the image onto this white board right here. Just set it up here, and I'll show you what we see when we look at this cellophane wrapping. Without the top polarizing lens into position, it looks just like plain cellophane. When we move it into position, we start to see a variety of colors that are given off by the polarized light. And as I turn this little polarizing lens, we start to see some really beautiful things. Remember that when you're doing polarizing experiments, that you use both of the polarizing lenses together. And when you're doing other microprojector experiments, you keep the polarizing filters out of the way and you don't use the P position on this lower turret. This next activity or demonstration is going to be using in the A position or amplify. And I'm going to use the 6.5 or the high power objective lens. We want to demonstrate to the students that cells really do exist. So we want to look at plant cells and we want to look at animal cells. The best type of plant cell to look at is the onion skin cell. And very simply, all you have to do is cut a little wedge out of a piece of onion and then carefully peel off a thin piece of the membrane. So thin that it's almost transparent. And I've peeled one off here, and you can see, if you look closely, that this piece of onion skin is very transparent. Break off the little piece of membrane and lay it on a slide. And then you want to stain the cells. They're much easier to see. One of the best stains for looking at cells is iodine. If you can't find it in the drugstore, buy it from a science supply house. Add 1 drop of iodine to the onion skin. And then you should cover it with what we call a cover slip. These are tiny little rectangular pieces of glass or plastic. I recommend using the plastic ones, and of course you can get them from the science supply house. When you put the cover slip on the slide, you always put it on at an angle first and just let it fall. When it falls, it spreads the liquid underneath it out evenly. Now let's take a look at this onion skin slide. First, your students should discover that this onion skin, this plant, is really made of thousands of tiny little cells. And secondly, they may ask you, what are all the little red dots found in each cell? Well, this is the nucleus, the central part of the cell. And you've been able to identify the nucleus by using the iodine because it's stained it red. If you look closely at this cell, you'll also see that the cell has a hard outer wall. This is called the cell wall, and it's found only in plant cells. Now let's take a look at some animal cells. And what better place to get animal cells than from a human? Get a volunteer from your classroom and have the person come up and give them a flat toothpick and reach inside in the inside of their cheek and scrape the toothpick along the inside lightly, like this. You'll be scraping off cells, very loose cells, from the inside of the cheek. These are called epithelial cells, and they're animal cells. You want to smear this on the slide, and then once again add a drop of iodine to stain the nucleus. Now let's see what these animal cells look like. Oh, don't forget the cover slip. By using the mechanical stage, I can turn the knob and move the slide around and hunt for particular parts of the slide that might be best to observe. What we're looking at here, besides these bubbles, this is an air bubble right here, but we're looking at hundreds of tiny little cheek cells. And if you look closely, let's see if we can find one to see look for a nuclei. Here's one right here. Let's take a close look at this one right here. You'll be able to see the nuclei inside the cell. Also, these cells, animal cells, don't have the hard cell wall that the plant cells have. You've just seen 3 applications of the microprojector, and there are many, many more. The bottom line is that this piece of equipment is versatile and very valuable, something your school should have, something that you shouldn't be afraid to operate because it's simple. And by all means, let your students come up and practice focusing and turning the objective lenses and so on. We will be using this piece of equipment throughout the course as we investigate many more tiny little things. With that last activity, it would be a good idea to have your students make good observations and drawings of the cells that they saw. Make comparisons between the plant and animal cells. Once again, we're working on process skills. Now we're going to continue by showing the students a model of a cell. Students should be aware that there's many different shapes and sizes of cells. Some are very long and resemble branches of a tree, some are round and some are rectangular and some are circular. What we're going to use for this model is a plastic bag that's filled with water and a water balloon inside. This is a very good representation of a cell. It has a hard outer layer. It's filled with an inner liquid, which we call cytoplasm, which is mostly water, and it has the central controlling part, the nucleus. Now it's like a little chemical factory. It knows what to do and when to do it, and it has the energy source to make new materials and get rid of wastes. And a cell has a special ability to reproduce itself, to make another one that looks just like it, or to make a diverse cell, a different cell. And this is how we all started, from one little cell, and we turned into the people that we are. Now let's go on to model number 2 and show how cells divide. This model, we're not using water this time, but we have a bag within a bag, and inside here is a secret message. You can hold it up to your students and say, looks like a little message in a bottle. Let's take it apart and see what's inside. When you open the bag up and you pull the little message out, you'll find that inside the nucleus there's a secret code. And in this code are all the instructions necessary and the materials for this cell to make a copy of itself. The first thing it does is it makes a copy of the code. So we now have 2 copies. And then it splits. So we now have 2 codes. And then it hunts around and it finds a nucleus for this cell, and it goes inside this bag, and lo and behold, we have another cell that's been put together here, and another little bag for this one. And this is the way cells divide. This is actually one way, and it's called mitosis. It's when cells replicate themselves and they split up and they form 2 new cells. And eventually this will happen between these 2, and when they split, we'll have 4 cells. And on until we start building multicellular plants or animals. Now, since not all of our cells are the same type of cells, there's a special thing that happens every now and then, and the little factory knows just how to do it and when to do it. And that little thing is to make new diverse cells, to make cells that are nerve cells or muscle cells or blood cells or skin cells. And the diversity of cells in an animal like a human is just fantastic. There's so many different kinds of cells. The size of cells are different and the shapes of cells are different. Some of the smallest cells are the bacteria, very, very tiny. And the largest cell is the ostrich egg yolk. So egg yolks are the largest cells. This next activity, we'll take a look at a chicken egg yolk. So we'll be looking at a very large cell. This is an activity you shouldn't pass up. It's great for developing process skills and only uses a dozen eggs or so. Before we get into it though, let's discuss how a chicken can possibly make one of these things in the first place. To do this, I'm going to use a model. I've taken just the yolk of an egg and put it in this olive jar. Now if you look closely at the yolk of an egg, you see there's really two parts. There's the nutrient part, which is the yellow yolk, and then on close inspection you see a tiny little white dot. This is where the chick will develop if the egg is fertilized. It takes about 9 days for a hen to make one of these yolks, and the yolk is deposited in a tube called the oviduct. It travels down through the oviduct much like it would travel down through this olive jar, and it gets to certain glands. The first gland secretes a chemical called albumin. This is the egg white. Traveling further down, it gets to the area where the shell is secreted, first as a soft material and then the shell hardens. The albumin is such that it holds the yolk of the egg directly in the center of the shell, so it's not leaning against the sides of the shell. And then of course the chicken lays the egg. Now if the egg is fertilized, this is a special kind of cell. It doesn't have the complete code like we talked about in the last activity. It only has half the code. And of course the sperm cell has the other half. And if the egg is fertilized, these two codes come together and then complete the code for the new chick to develop. And you can go back to this and cut your code in half to explain this. And it's a good lesson in reproduction too. Special cells have only half the code, therefore the male part can come in with a female part and make the new offspring. Now let's get on with this egg observation activity. What are some of the things we can do with the egg before we crack it open? Well, first, your students can record the shape. Make a drawing of the egg and notice that it's flat on one end and kind of pointed on the other. You could have them weigh their eggs and find out which group has the heaviest egg. You could have them measure their circumference around using a piece of string and wrapping the string around the circumference of the egg and then measuring the length of the string. Or you could use sizing squares like we used in earth science and see which size square the egg will pass through. You could also have them get out the magnifiers and look for distinguishing marks on the egg and record this information. And then you can take their eggs away and mix them all up and see if each group can identify their original egg. Now we're ready for another experiment. This time we're going to float the egg in a jar of fresh water and make observations. If you've chosen an egg that's been out of the refrigerator for a few days, you'll see that when it's placed in the fresh water, it has a tendency to float with the rounded end higher than the pointed end. It sits up almost on one side. Why is this happening? This is a question you want to ask your students. What do they know about things that float, and why would it have a tendency to float with one end up? Well, maybe somebody will propose a model. If not, you might help them along. This model I'm using is a little jar with an air pocket in it. And when we place the jar in the water, we'll also see that it floats up where the air pocket is. In this case, though, if we turn the jar upside down, it'll also float upside down with the air pocket. We could test that idea with our egg by placing it with the pointed end up and see if it floats that way. In other words, does the air pocket move in the egg? And we'll see that that's not the case. It has a tendency to flip back around and point in its original direction. So the conclusion that we must come up with from this model and the observations from this experiment is that there's a little air sac inside the rounded end of this egg, and it's kind of stuck in there. It doesn't move around inside the egg. And we'll look for this air sac when we crack our egg open. Another thing we can do is what's called candling. And in this experiment, your students can get flashlights and put little tissue tubes on the end with tape. Darken your room once again and shine the light into the egg. And students can look into the egg and see inside the egg. They may be able to see the yolk. They should be able to see the little air pocket, and of course they'll be able to see all the imperfections in the shell. Places where there's been an internal crack in the shell and the chicken has actually sealed it up, much like we seal up bones when they get cracked or fractured. Now let's go on and crack this egg open and look at the inner parts of the egg. There are 5 main parts to look for. The first and easiest, of course, is the egg yolk. Inside the yolk is the little tiny white spot, or germ. That's where the chick develops. Connected to either end of the yolk, there's some little white thread-like material. This is called albumin, and it's used to hold the egg yolk in position. Further out from the yolk are 2 other types of albumin. One is very thick, and the next one is much thinner. The relationship of the thick to the thin determines how fresh the egg is. And of course you want an egg with more thick albumin than thin albumin. When your students look inside the shell, they'll see just what they expected, a tiny little membrane with an air pocket inside. This is what was making it float with the rounded end up. This air is also used by the chick a day before it hatches. Now that your students are all experts on eggs, we'll do one final experiment to see if they can apply this new knowledge. This time we're going to take one egg out of the refrigerator each week for 3 weeks and mark it with a crayon the date it was taken out. It can be done at home or in the classroom as a demonstration. After 3 weeks, we'll have these 3 eggs. We're going to make some guesses about them. The first guess will be, which one will float the best? Which one will float the highest in the water? Well, the theory is that as the egg sits out, the liquid evaporates and more air appears inside, so a very old egg will actually float to the surface. We'll try that experiment and see if we were right. The second experiment we'll make a guess about is when we crack them open, what will the albumin look like? Will there be a lot of thick albumin in the old egg or in the fresher egg? We'll make a guess about that. We'll crack the eggs open and we'll take a look. If your students do this at home, have them crack all their eggs the same evening and report their statistics back the next day. As we continue to study life, there are other activities that we can use to help reinforce some of these important concepts. When we go back to genetics, you can make little puzzle cards like this. They not only show how a boy turns into a man and how a tadpole turns into a frog, but it also illustrates that a boy cannot possibly turn into a frog because of his little genetic code, the particular chromosomes that he carries within his cells, and they're different from those of a tadpole. This is a great little activity for getting this simple idea of genetics brought out. Another fun thing we can do is try and identify things as living and non-living. And in this activity, have your student groups of 2 or 3, go around from station to station, look at an object, and record whether it's living or non-living. And I've got some little tricks here. For example, here's a pencil and a golf ball embedded in potting soil. At station number 4, our students will find a bug box with a magnifier in the top, and this one has a dead insect in it. What about lima beans? Well, they're living. They only require the proper conditions to begin to grow. They may be considered life in a resting state. Some other good examples you might try are a battery, and you can always use little vials and put an insect inside. Remember, there are 5 characteristics of living things. They need food, they move, they grow, they reproduce, and they're made of cells. As your students get more involved with the study of life, they should learn to classify both plants and animals according to a variety of characteristics, but only considering one characteristic at a time. For example, animals might be classified according to what they eat. Are they meat eaters, plant eaters, or do they eat both? What type of body covering do they have? How many appendages do they have? And where do they live? Your students might classify plants according to what the bark looks like, or whether it has bark or just a stem, or what type of roots does it have, or does it have flowers, or berries, or nuts. Another thing students should learn is that there's a great diversity among living things, many varieties of plants and animals. And there are 3 broad categories that we classify all living things. They're plants, they're animals, or they're what we call protists. Protists fall into a kind of a hazy zone between plant and animal, and many of them are unicellular, which means they're one-celled animals. As we look in our microprojector, we can look at some of the simplest forms of life and some of the protists at the same time.